bunch of, yeah. Thank you, JC. Yeah, and I thought it was really good. It's actually a great introduction to Fermi estimation in general. They even have like sections like kind of summarizing how to do order of magnitude estimation and stuff. So I think like the best people at order of magnitude estimation are like astronomers and maybe biophysicists. So this is like a great way of getting into there. And I think a lot of people are interested in that. The other thing I know a bunch of people are interested in is like climate stuff and global scale, energy fluxes and CO2 fluxes. So we'll talk about that too. So yeah, so the main idea here, I'm going to do it a little differently than before. I used to kind of do a bit of a lecture where I went through some numbers and then gave you some practice and then did a big one where we did breakout rooms. But I was thinking now I wanted to give you guys a chance to guess some of the numbers and quantities before I tell them to you and work through some answers. And I think that will both help practice the like boldness that I think is one of the hardest skills to learn here, which is taking anything you know from like any part of life that could be partially relevant and using that to constrain, guess, get like some rough handle on what things could be. And I think practicing that is really good. And secondly, if it turns out that like, you didn't know the answer, then when you see the answer, it's more valuable, more surprising, and will stick with you better. Alrighty, so let me get my slides up. And let's share the screen.
You can take the co-hosting away from me if needed and give it to iPad. That might be that. Oh.
Okay, so can everybody see the slides? Okay, so yeah, today we're going to be focusing on E. coli. There's a lot of great stuff, which I'll talk about a little bit more. But first thing I wanted to highlight is actually this picture. So this is by David Goodsell. He has an amazing book called The Machinery of the Cell, and where he goes through also a lot of these kind of order of magnitude parts of the cell. And what's really cool is he runs this like protein structure database that's been going on for years and years that has a lot of protein structures. And he also makes these amazing physically accurate like paintings of cells. So he made this painting here of E. coli, and he tried to get the right densities and sizes of various molecules here. You can see, I think the yellow is kind of like DNA. The green is like your lipid membrane bilayer. You can see at the top left there kind of a flagellum. And one of the big things you can notice is like how crowded stuff is inside of the cell. That's like often very surprising. I think this, one of the cool things about order of magnitude estimation is that this is a regime where maybe we don't have a lot of like intuitive experience, but we can take knowledge we do know and kind of like, scale it down to this size. And I think also by having pictures and by getting some of these rough estimates, we can start to build some intuition for, what cells feel like and what their worlds are like. What I wanted to talk about today was we were going to use E. coli as our like standard, which is a prokaryotic cell, so it's much smaller than like our large eukaryotic cells. But we only have so much time, and to be totally honest, I was in a bit of a rush to make this, so sorry about that. So first thing is we're going to try and estimate some sizes here, some volumes. How big is an E. coli? How much does it weigh? And then we're going to go to some time scales, like how fast is DNA transcription, for example, based in like how fast does E. coli divide? And yeah, so I got a lot of these numbers and pictures from this book, Cell Biology by the Numbers. So Physical Biology of the Cell is this huge textbook, hundreds of pages, very dense, but really awesomely, they also made a much more like simpler, pared down introduction called Cell Biology by the Numbers. And there's a draft of the book that's free online and a nice website. So if you guys like, want to take a, read through a book and like absorb a lot of great knowledge, at a kind of deeper pass, but really accurate, this is where to go. This Cell Biology by the Numbers, I highly recommend. Just something to point out on the textbook cover, they do a fun thing of like, making this map where they name all sorts of different, non-polar region, potassium current and stuff. So that's kind of fun. Okay, so we're going to start here with E. coli. And the first thing I want you guys to estimate is what is the length of an E. coli cell. So I'm eventually going to tell you to move on to the next thing, but this is to practice, getting some estimates. Maybe you have no idea, but like, do you think it's like an angstrom or a nanometer? Do you think it's like a millimeter or a meter? Do you think it's a micron? And like, are there any other length scales that you can kind of use to bound that? So why don't you all take a minute or so and give your best guess, and maybe try and think about different knowledge that could help link that up for you. So Venkat made a great point. E. coli can be seen with a light microscope. And I think it's probably near like the bottom of what a light microscopes can see. So if you're a little stuck here, that might be a great like thing to anchor on that could maybe help you figure it out if you know something about wavelengths of visual light and stuff. So thanks, Venkat. All right. So I think it's about time to see how people figured out. So maybe first in the chat, maybe if people had guesses, they could put it in the chat. We could see kind of what the range was of guesses. All right, I'm seeing David White says that light microscope resolution is about 200 nanometers, so he'd guess 2 to 5 microns. Ben said 1 micron. So these are great guesses, and you're right. We're going to kind of guess that the diameter is about 1 micron and the length is about 2 micron. And so one of the things in the cell numbers book, they have this nice log scale here of different sizes of things. So, water molecule or glucose on the order of 1 to 10 nanometers. We know from previously atoms are on the order of an angstrom, which is 1/10 of a nanometer. So we have like, atoms, molecules, slightly bigger molecules. Yeah. And then now, we're getting to electron microscope resolution. You can't really see like these teensy molecules, but a little bit better. Then we're getting up to the size of proteins, ribosomes, viruses. And here we are, just like David White was saying, light microscope resolution at about 2 to 500 nanometers. And at 1 micron, we have E. coli. And then keep getting bigger and bigger here, we get all the way to, this must be like 10 to the 7. So maybe that's like 100, 9, 8, 7, 6. Yeah. So this is order 10 microns, 20, 30, 50 for eukaryotic cells. So now we have 1 micron. The next thing to guess is the volume of an E. coli cell. So this is, another thing to practice. Now we have this number, right? A micron is 10 to the minus 6 meters, 1 millionth. And so now this is practicing a little bit more, doing that, the like powers of 10 scientific notation stuff, and just practicing with that, going from lengths to volumes. And so we'll see how that goes. And JC asks, what was visible to the naked eye? Interestingly enough, I would guess something like, the length of a width of a human hair is probably the like minimum resolving thing for eyesight. And I don't know off the top of my head, but I would guess that's between 10 and 100 microns. So yeah, it's like I think you could see kind of large eukaryotic cells. I think E. coli is like, smaller than that, but yeah, that's a good thing to anchor on if you did know that length scale and kind of compare around. You're definitely getting in the micron range there. So that's about, decently good size. All right, so yeah, I'm seeing some people having answers here. I'll give a few more seconds, and if other people want to put their guesses in the chat, that would be great. Yeah. And again, one thing I want to remind people, particularly those that are joining us for the first time, we are doing Fermi estimation or order of magnitude estimation. All we care about is if we're within a factor of 10 from below or above. So if the answer is like 30, like between 3 and 30 counts, and between 30 and 300 counts. So you have like an order of 100 like total thing. And like that all is good. We're just trying to get within an order of magnitude of the right answer. So like definitely, feel free to, if you feel confident on, putting some numbers in there, that's like great. That helps get you like cancel out some factors of 10, but like we're really being rough here. We're being fast and loose. So, okay. So this one, pretty straightforward. As I said before, you could think of this as like a sphere or a cube or a cylinder or a rectangle, but it's going to be about 1 micron on all 3 sides. So the length is a micron, the volume is going to be that length cubed. And so if 10 to the minus 6 meters of length, we're going to get like a cubic micron, or 10 to the minus 18 cubic meters, 10 to the minus 6 cubed. So that 6 multiplies the 3. Minus 6 times minus 6 times minus 6. So that's great. So now this is the fun thing. I don't think we really need to like know the volume that much, but something we do want to know is what is the mass of an E. coli. And the thing I want you to be thinking about here is that the density of water is 1 gram per cubic centimeter or 1 gram per milliliter. And so using that, and we have this volume from before, you should use that to figure out, yeah, the mass of a E. coli cell. Yeah, Lewis, the density of water is 1 gram per cubic centimeter, a gram per cc. And you know, I think that also works out to like a kilogram per liter, but I'm not 100% sure. You know, I think these are like, you know, base things of the SI unit system that kind of like, you know, everyday human amounts of water and human weights and human lengths. Yeah. All right, anybody, any other guesses here on mass of a E. coli in the chat, perhaps? All righty, well, we had that volume, 10 to the minus 18 meters cubed. And if it's a gram per cubic centimeter, then just like we saw in the chat there, I think that's 1,000 kilograms per meter. Is that right? Gram per cubic centimeter. That's 10 to the minus 6 because it's 10 to the minus 2 cubed. So yeah, 1 million grams per cubic meter. And yeah, and we have 10 to the minus 18. So 10 to the minus 18 and a 10 to the 6 gives us a 10 to the minus 12 grams for the weight of an E. coli. And that is a picogram. So that's the right mass scale there, is a picogram for an E. coli. So just to review, 1 micron is about the size, a cubic like micrometer is about the volume, and a picogram is about the mass. Great. So next thing, this is kind of fun. Just you know, interesting question also from the book. Which is bigger? So going back a little bit, how does biology work? What is this central dogma of biology? And the central dogma is that you have DNA, which is like the recipe book. DNA holds the information. You don't want to use the whole recipe book. Sometimes you copy down a recipe, which is like a written page or a shopping, like a grocery list. So you translate out the coding sequence you want to make into a protein. That's mRNA. So that comes out of the nucleus. And then the mRNA is read by the ribosome, which then takes that information and uses it to attach amino acids together that then folds into a protein. So the central dogma says DNA to RNA to protein. And you may or may not remember or know that there's this thing, the genetic code. So normally when you hear genetic code, you think like DNA and the information in there, but that is not what genetic code actually refers to. Genetic code refers to the 3 base pairs of an DNA or RNA that code for an amino acid. So there's this 3-to-1 base pair to amino acid. So I think that maybe helps a little bit. So knowing, maybe you could guess what is the relative size of a nucleotide versus an amino acid, how many nucleotides per amino acids, stuff like that. And then or maybe also just think of if you have something that's information and something that you're building, how does that normally correspond? So yeah, so the question is, which one's bigger, mRNA or protein? And then maybe like how much bigger is one versus the other, or are they like about the same? Yeah. So yeah, that's a good question. I see both Venkat asking about amino acids versus nucleotides, and then David White giving like a 4 to 5 to 1 answer. So first, Venkat, maybe if you want to like think aloud about your question there, and then second, David, if you maybe want to like explain how you got around your answer.
Yeah, I vaguely remember from long ago that the alphabet is like 3 letters from the set A, T, C, G. So it's like 3 of those size molecules. I don't remember anything about the chemical structure, the adenosine or whatever.
Yeah.
But amino acids, I assume, have an amine group, so NH3, I guess.
That's right. That's exactly right.
Very vague old chemistry,
Mhm.
but I literally have nothing to compare it to on the other side. Like I remember nothing about adenosine structure.
Yeah. So so that's great. That's exactly right. So just for everyone, as I should have put pictures here, I apologize. But an amino acid is this like amine group. It's a nitrogen is the center. It's got 3 hydrogens on one side. Maybe there's a like carboxyl group stuck on there because it's an acid, so probably. And then there's this R group, and that's the thing that changes among amino acids. That's some like slightly larger little molecule, but order, I don't know, 10, 20 atoms, something like that. And then nucleotides are 3 parts. They're, I think, this like phosphate. They're this ring, this like carbon, maybe nitrogen ring that's either 4 or 5. That's the purine or pyrimidine. And you're always like pairing those. So A and T, C and G, one of those is a purine, one of those is a pyrimidine. And then, if you think deoxyribonucleic acid, so deoxy means no oxygen, ribo is like ribose, it's a sugar, and then like nucleic for the other part. So, I think it's bigger, it's much bigger than an amino acid. All right, so, and then David, what were you thinking a little bit about the 4 to 5 to 1?
Yeah, I mean, it just because the codons are, it's 3 to 1. And so that already gives you the ratio. And then like sugars and phosphates are relatively big compared to most protein side chains. There are some bigger protein side chains, but like that core amide structure and then the side chains on average tend to be pretty small. Like it's not like you're going to pull a whole bunch of tryptophan or something in a molecule. There's going to be like a mixture.
Yeah,
that'll be small.
So that was a lot of words. and a picture's worth 1,000 words. So here's the picture from the book. And you can see that the mRNA is actually like way bigger than the myoglobin that it's coding for. And this is like a regime we're not really used to, right? You're not usually used to like the blueprints for a house, like being way bigger than the house or something, right? Or like, your blueprints for a jet or like something, a toaster being bigger than the actual structure. But in this case, that's the case. Your nucleotides are bigger than amino acids. It's a 3-to-1 ratio. And, proteins also can like fold up pretty densely, and that mRNA needs to be more linear. Yeah, so pretty interesting result there that I didn't actually realize myself until I was preparing for this.
James,
and yes,
can you, I mean, is there speculation on, this is a process that has repeated lots of times? You could probably Fermi Gym a number of repetitions. Like, why is this so counterintuitive that we have this long plan and low results? Does it say anything about like information capture over long periods of time and many repetitions, or is it just like that's how it happened? How should we think about this kind of counterintuitive structure of information capture?
So that's a great question, and I don't know the answer. So this is me, ignorance never stops me. I'm going to just speculate wildly here. But my guesses would be several-fold. The first is that we want to one is that this needs to be a general mechanism, right? That you need to really be able to have something that can encode arbitrary sequences. And you have this general device, this ribosome, that it can read in like arbitrary kind of like words or sentences and output something. And so that in itself, that generality, I think requires some extra complexity. It turns out that there are certain kind of complex molecules that you want to make a lot of. So like fungi create these, often like antibiotics are created through this other process, which is called non-ribosomal peptide synthesis. So you'll actually build like other machines that can like pop out these molecules much faster. It's kind of like 3D printer versus like, injection mold or something, where you have like one thing you're making a lot of versus the like slower, more complex, more general thing. So I think that's part of it. The generality makes it bigger and more complex. The second thing is we want to persist information like robustly. And so that means you have like this 3-to-1 ratio. It also means that you want something that takes a lot of energy to make. So, molecules are, I think, fundamentally out of equilibrium. This is why like DNA and RNA degrade over time, right? So RNA degrades on like minute time scales. DNA degrades on like, I don't know, year time scales or less. But like the environment, the thermal environment of the cell, like if you're looking at thermal fluctuations, they're like decently big, and so to get something that stays out of equilibrium for a long time, you have to dissipate a lot of energy. You have to so you could think of like, cars want to rust, for example. And so we actually put a huge amount of energy to like melt the iron, turn it from rust into real iron, and like this huge amount of energy versus just like if you were perfectly breaking bonds. You put some extra energy and then we like fold it and we put a lot of energy into the car so that it like holds up against rusting and breaking apart.
Oh, now you want.
Yeah. So I think that's maybe another part of it, is to persistently store information against like out of equilibrium. I think you need like large, big, heavy and energy rich structures. So that might be another part of it, but that's just me speculating. But great question. Yeah.
No, thank you. It's fascinating. I mean, how much of our lives is compression, and this is the other way, right? Yeah. Anyway, thanks.
Does anybody else have other ideas or things they'd want to add or point out? on this process?
Just a kind of, this is not relevant to Fermi estimation at all, but since mRNA vaccines are in the news, I thought I'd mention that this has like upended my picture of what the vaccines are physically like. Like I had this vague, unfounded notion that they're a very small molecule that are being injected in us to produce protein. This sort of flips it around. So I now assume that mRNA vaccines are what we got injected is like big molecules, not small.
Yeah, that's a good point. I hadn't even made that connection myself. Interestingly, at the Cell Biology by the Numbers website, I haven't looked at it, but they also apparently have a COVID by the numbers. So I think they might, like, they probably go into that comparison in there. Yeah, and finally, I wanted to point out, just some numbers here. So 5 nanometers, I guess, is about the size of a myoglobin, which is probably like a kind of small protein. And that looks like it's only a few bases of this RNA. And for some other numbers, it looks like 1,000 bases, maybe codes for like this kind of small protein here. So a kilobase per protein for information. So with those numbers, we'll go to our like last questions of the day. First of all, I wanted to show you this, which is a real electron micrograph of DNA being transcribed into RNA. So, reading that recipe book and making the recipes. And then before the RNA like even comes off, there are ribosomes sticking to it. And those ribosomes are reading the RNA sequences and having like proteins start getting made off of them. So, I would have thought, oh, there's like RNAs floating around and maybe and like the ribosomes may be bigger than the RNA and like, it reads it one at a time, but like apparently no, like ribosomes are smaller than the RNA things. And there's like a whole bunch of them all reading it at once, like kind of insane. And to note the scale here, before this was 5 nanometers and now we're at 500 nanometers for this scale. So some other questions. And I don't know. Probably they did a lot to make. So David White was asking, is there a contrast agent here to see this? And my guess would be probably. Usually my understanding. Well, I don't know. Probably, I don't know. This is from the Cell Numbers book. So I think in their description of it, which I didn't copy, they talk more about this picture. Okay, so this final thing is about speed of speed of DNA transcription. So like how fast are these transcriptases moving along the DNA? How fast are the like ribosomes moving along the RNA? What's that time scale? So the first thing, the way we're going to kind of wrap our estimate this is we're going to try and estimate 3 numbers. We're going to first say how fast does it take like E. coli to divide as a whole, and then how big is the E. coli genome? And if we think like most of the division time is like copying that DNA again, then that might give us some estimate for how long it takes like per base pair to copy. So the question really here, we're focusing on like how long does it take to copy all the DNA in E. coli. So I'll give you a little bit of time to try and guess some of these questions. And it's okay if personally, I wouldn't really know exactly where to start on this. I would just guess some numbers, but I do know that the human genome is on the order of like billions of base pairs, gigabases, and E. coli is like smaller than that. And I know human cells take like many hours to divide, and so if E. coli is less than that, take that information in. So anyway, I guess like do your best, get some guesses here of what you think it might be. Alrighty, so I see some guesses for the first one, how fast E. coli divides. And I apologize, you know, the books give some like ways of estimating this a little bit better and give some scaling relations. I didn't actually like read that in depth and like synthesize it so I could pass it on to you in a nice way. So we're gonna kind of just get this as, one of those numbers to know for now. But the answer is it's about an hour. It's like order 3,000 seconds, and an hour is 3,600 seconds. So it takes about an hour for an E. coli to divide. And that's kind of as fast as, bacteria go. They're small, they divide pretty fast. So that's like kind of a lower bound on how fast prokaryotic cells or all cells can divide. So that's that one. So I guess next, for people, to kind of guess how big the genome is, how many like nucleotides do we need to read through in that hour length of time to copy everything in time. Yeah, so Lewis asks, do we know how much larger human cells are versus prokaryotic cells? And we do. I would say order 100 times larger, although there's like a fair amount of variation, eukaryotic to prokaryotic. And the interesting thing to remember there is, so there is a scaling relation between size or volume and division time. The big like innovation of eukaryotic cells versus prokaryotic cells is prokaryotic cells are like limited by the surface area to volume ratio, right? Like a lot of their stuff happens on their surfaces, like having the ATP synthases, they're on the surface. And so you have to have some proton gradient that drives things, and their DNA they don't have a nucleus, so it's just like in the cell. The cool thing about eukaryotes is because they have organelles, you can decouple like your surface area to volume ratio. You can get much larger, but then since you have these like modular subparts and they have their own membranes, like they can still have the right surface area to volume ratios to have their processes going, and yet you can make the whole cell like much bigger. Yeah, so that's like the main idea there of like why eukaryotes are a big deal is because they have internal membranes, you can like not have to just have everything based on the surface area of the whole cell. Yeah. Yep. Yeah, everybody's saying great stuff. So thanks for thinking hard about this and linking up various things. I think, as several people have pointed out, like being able to see like by your eye on like millimeter scale, growth of bacteria on the order of like a day or two is like a great way. We know micron size for a single one, and order 100 doublings to get to millimeter size. And so like how many doublings would have to happen in that case? That's a great way of bounding the problem. That's the exact right thing to do. So I wonder if you like took those numbers, if it would work out right. For the E. coli genome, I was seeing hundreds of kilobases. That's close. Apparently the answer is on the order of like 1 to 5 megabases. So 5 or 6 million base pairs for the full genome. Several orders of magnitude, like 1,000 times smaller than humans. And so now we have these two numbers. We have time to divide 3,000 seconds. We have how many total base pairs do we need to do in that time? 1 million. And so now quickly estimate like how fast does it take to read like one base pair? All right. I'm seeing some answers on the order of 10 to the minus 3, a few milliseconds. So like 1 nucleotide per 10 to the minus 3 seconds. And I was reading through the book real quick, and the scale they were looking at was how long does it take to read 1,000 kilobases? And I guess the rate is about 100 nucleotides per second. So that's about 10 milliseconds per nucleotide. So close. Everybody was pretty close, within an order of magnitude, I think. And yeah, so then 1,000 nucleotides in 10 seconds. So that's the scale there. I think my guess is that eukaryotes have about the same speed, and we just have 1,000 times larger genomes. So instead of like order hour, it's 1,000 times more. It's order 1,000 hours.
A question here.
Slow.
I assume there's a lot of parallelism
Yes.
in the ribosome level. So what's the speed of a single transcription? That must be much slower, right? Because there's like 100 ribosomes, maybe, I don't know.
That's exactly right. So what we're, I think this 80 nucleotides per second is the speed of transcription. And you're right, it is massively parallelized. So that's like a great decoupling there. So my guess would be if you just took this 1,000 base pairs in 10 seconds, then 1,000 times more would, that's about 10x too large. Yeah. So that's interesting. I don't know why there's not more. It's because maybe that usually there's copies, but like usually there's like a single circular genome for E. coli, and humans have a bunch of different chromosomes, right? So like you need to make one long sequence. I don't know if you can, do it multiple times and then stitch them together. I'm not sure that works. I think you just have to read the whole thing. And so there's not that much parallelism you can achieve by having different parts of DNA and then attaching them together. So that's interesting. There's that like fundamental limit to parallelism there. But if you have multiple chromosomes, you can like do all of those in parallel. So yeah, great. Never thought of that before, but that's an interesting point. So that was kind of the stuff I wanted to cover. I'm happy to like answer any questions and stuff, but we're kind of, I think we're about done. And so yeah, so I hope you guys all enjoyed it. And next week, I unfortunately will be traveling to back from Austin for a company all hands. So won't be available next Saturday, but Saturday after, and then I think going forward for a while from then, we'll have it every Saturday. So great to see regular people. Thank you for the new people that were coming. Yeah, really appreciate you all taking the time out, and happy to chat some more.